All right, this is our second video on cell physiology. In the first video, we got kind of an overview of cell physiology, and then we talked in some detail about the structure and function of the plasma membrane. And again, I want to remind you that the plasma membrane is not the same as a serous membrane, which we talked about when we covered body cavities and the covering of organs within the body cavities. I also want to remind you that the structure and function of the plasma membrane is considered a key concept in physiology, and so you need to make sure that you understand those well. Okay, in this video, we're going to start to talk about the other parts of the cell, including the nucleus and the cytoplasm and what we find inside the nucleus. Okay, one question you might ask is, do all cells contain a nucleus? At any rate, I'm going to ask it. And the truth is that there are some that don't. And the best example of a cell without a nucleus would be a mature red blood cell. The reason for this is that the nucleus, along with other organelles, is kicked out of the red blood cell so that we can put in as much hemoglobin as possible. The job of the hemoglobin is to carry oxygen that you need for cellular respiration, and we want it to hold as much as possible. In this diagram, you can see the development of a red blood cell, and it does at first have a nucleus. But as it develops, at some stage, it actually pushes the nucleus out, along with other things that are in there. And this allows us to pack in lots and lots, indeed millions of molecules of hemoglobin. Some cells actually have more than one nucleus, and so skeletal muscle fibers are what are referred to as multinucleated. Cardiac muscle cells might contain only one nucleus, but on the other hand, some of them contain 2 or even 3 nuclei. And then bone osteoclasts are also multinucleated. We'll talk about osteoclasts and what they do more later on in this semester, but here you can see an osteoclast, and you can see multiple nuclei inside that osteoclast. Let's go ahead and look at some more detail in terms of the structure and the function of the nucleus. You know that the nucleus is oftentimes considered the brain of the cell. Here you can see a nucleus. It almost looks as if it might be the whole cell, but no, indeed it isn't. This is the nucleus. And if you look carefully here, you can see one of the distinguishing characteristics of a nucleus is actually a double phospholipid bilayer. And this is referred to as a nuclear envelope because it has an outer nuclear membrane and it has an inner nuclear membrane. And here you can see a bit more detail. Here's the outer nuclear membrane. This is phospholipid bilayer. Here's the inner nuclear membrane. It is also phospholipid bilayer. Well, what is it that we have inside of the nucleus? Well, what we have is we have protein, DNA, and RNA. And you know DNA and RNA are nucleic acids. Most of what you see here is really DNA that's really spread out and diffuse. And it's in a form that's known as chromatin. Chromatin is DNA that's very diffuse, and it is wound around proteins that are known as histone proteins. And we'll look at that more later on when we talk about mitosis and cell division. The other thing that's happening in here, we also have this structure in the middle known as the nucleolus, and this is where transcription of ribosomal RNA is occurring. Another important structure in the nucleus are these nuclear pore complexes, which are sometimes simply referred to as nuclear pores, which really isn't a good idea because, as we'll see in a minute, these are more than just holes through the nuclear envelope. A nucleus has somewhere around 3,000 to 4,000 nuclear pore complexes. And then we also see that it's studded with ribosomes. To be perfectly frank, I don't entirely know what the job of the ribosomes that we find on the nuclear envelope is. Maybe we'll learn that someday. We have lots of things that will pass through the nuclear pore complexes. Some go through passively. Examples would be ions, so sodium, potassium, those types of things, and also small molecules. Other things, on the other hand, pass through actively. And you know that means that we have to use energy in the form of ATP for that to occur. Things that move through actively include protein and then also RNA or ribonucleic acid. Things that move into the nucleus include nucleotides, and that shouldn't surprise you at all because we need nucleotides in order to build nucleic acids, DNA and RNA. And something that moves out would be RNA. And in particular, messenger RNA is going to move out through the nuclear pore complex. Here you can see a bit more detail for that nuclear envelope. And again, here's the inner nuclear membrane, and here's the outer nuclear membrane. And then here they're blowing it up to show you that each of these is indeed a phospholipid bilayer, 2 layers of phospholipid. And again, here you can see the hydrophilic heads of that phospholipid bilayer, and here you can see the hydrophobic fatty acid tails. This is also a phospholipid bilayer. And then here you can see some nuclear pore complexes that pass through that nuclear envelope. You can see that. This shows you a little bit more detail. So here we look at the inner nuclear membrane and look very, very closely, and you can see phospholipid bilayer. Here's the outer nuclear membrane, also phospholipid bilayer, and I do want to point out to you that it continues outward and it actually becomes the membrane of the endoplasmic reticulum. And we'll talk more about that later on when we start to talk about organelles of the cell. Here you can see a nuclear pore complex. This nuclear pore complex, again, is more complicated than just simply a channel through the nuclear envelope. I'm not going to expect you to memorize this structure. I just want you to know that it is somewhat detailed, and there are lots of proteins in here, some of which work as transporters. So that's really what I want you to know. And again, here you can see inner nuclear membrane, outer nuclear membrane, which are both phospholipid bilayers. So I hope that I've beaten that horse to death enough for you. Again, another part of the nucleus is this nucleolus, which is found inside of the nucleus. The truth is that a cell could have anywhere from 1 to 4 nucleoli. And so the more protein that a cell makes, the more nucleoli we're going to find, the larger they're going to be. I want to reiterate that the nucleolus is non-membrane bound. There is no phospholipid bilayer here. It's made up of protein and nucleic acids. I also want to point out to you that the nucleolus is not found in the cytoplasm, so technically the nucleolus is not an organelle. It's part of the nucleus. And again, what happens here is we get transcription of ribosomal RNA, and this is where we manufacture those subunits that are going to form ribosomes. We'll talk about those ribosomes later on. We'll also talk about transcription. Many of you remember from biology class what transcription is. We'll go over that. If you don't remember very well, or if you didn't cover that, we will talk about that in class. And again, cells that produce lots of protein will have large nucleoli and will tend to have more than one of them. All right, now we come to the cytoplasm. The cytoplasm is everything that we find outside of the nucleus, but inside of the plasma membrane. So here's the cytoplasm. And a large part of that is really fluid in nature. And so the cytosol is the fluid part of the cytoplasm, and it's oftentimes just simply referred to as intracellular fluid. We'll talk about body fluids in this class, and so intracellular fluid is an important component of body fluids. And then we have organelles, or little organs. Years ago, we used to say that the organelles were membrane bound. We've expanded that, and we'll see that there are some exceptions to that. And then inclusions are those things that are in direct contact with the cytosol. In other words, they're not membrane bound. And many books will include cytoskeleton and ribosomes as inclusions, although more and more we see books moving away from that. And our book and other anatomy and physiology books tell us that the cytoskeleton and the ribosomes are organelles. It's really not a big deal. You just need to understand structure and function of these things. And then organelles and inclusions are not cells. Sometimes students in anatomy and physiology tend to talk about the organelles as if they were cells themselves. They're actually parts of cells. They're not cells. Cytoplasmic inclusions are found in the cytoplasm, and they could be things that are produced by the cell, or they could be things that are taken into the cell. The cell produces lots and lots of things that are made from protein. And so we've already mentioned hemoglobin, and you know hemoglobin is a protein that carries oxygen, and so it's found primarily in red blood cells. Melanin is a protein that helps to give color to certain cells, and so cells with lots of melanin will be darker. And then enzymes are also protein, and cells will have thousands of enzymes that are free in the cytoplasm and help to catalyze reactions. Lipid droplets and glycogen granules are things that are taken in from the outside, and they provide an energy source for cells. A little bit later, we'll talk about cellular respiration and how we get energy, and so then we'll talk about the breakdown of lipids and glycogen to get energy. Organelles, again, are little organs, and traditionally these were things that were membrane bound by a phospholipid bilayer. Exceptions to that include the cytoskeleton, ribosomes, and centrosome. All of those are non-membrane bound. There's no phospholipid bilayer associated with these things. The number and type of organelles that we find in a given cell are going to be related to both the structure and the function of the cell. And so some cells are going to have a lot more cytoskeleton. Some cells are going to have a lot more rough endoplasmic reticulum. Some cells will have a lot more smooth endoplasmic reticulum. And you'll be able to determine that from knowing structure and function of the cell. Let's start by talking about the cytoskeleton. Here we can see the components of the cytoskeleton. The cytoskeleton is made up of 3 different types of protein filaments. The largest of these protein filaments are the microtubules. They're called tubules because they do have a lumen or an empty area that runs through the middle of it, much like you would find with a straw. And here you can see protein globules that go together to make up these microtubules. Microtubules form cilia that are used for movement on the surface of cells. They also form the flagella that we find in the sperm cell. And they also form a vehicle transport highway within the inside of the cell. And so when we need to move something from one part of the cell to another part, it can travel along the surface of the microtubule, and this might surprise you a little bit. You might think, oh, it's just going to travel through the lumen, much like blood travels through the inside of a blood vessel. And that's not the case. It actually gets dragged along the surface so that it can get from one part of the cell to another part. Moving up in size, we move to intermediate filaments. Here you can see that there are several protein fibers that are twisted around one another, much like a Twizzler. And they generally provide a supporting framework for cells, and they also provide some structure to hold the organelles in place. Here you can see some intermediate filaments that are holding ribosomes and also mitochondria where they need to be. In neurons, they form these long fibers that run down the axon of the neuron, and that helps to provide a little bit of stiffness for the axon and give it some direction. And then your skin cells, the epithelial cells that make up the skin, have keratin intermediate filaments, and they help to provide some toughness for those cells, and also they help to tie them together to make a tissue where the cells don't pull apart easily. The smallest of the cytoskeleton elements are microfilaments. Microfilaments are basically actin, and so that's the protein that makes up these guys. And here you can see little individual globular actin that have built into these microfilaments. They help to provide some stiffness for the cell, and so it's not quite as fluid as it might be if they weren't there. And some cells have to move. And so you know that some cells are phagocytic. They can reach out and engulf things. It's the microfilament that allows the membrane to do that. Other cells have to move from inside the cardiovascular system to out into the surrounding tissue. And again, it's actin that allows those cells to change shape and to squeeze through the cells of the blood vessel to get out into the surrounding tissue. Here you can see another depiction of those elements of the cytoskeleton. Here are the microtubules. They're the larger of those elements. Here you can see the intermediate filaments, and then finally the smallest filaments, the actin filaments. The cytoskeleton has several functions. As we've already said, cytoskeleton can provide some shape to the cell. Here you can see that it does that, and also some organization. And so those fibers can help to hold organelles in place. Here you can see a mitochondria being held in place by those things. They also provide some transport within the cell. Intracellular means inside the cell itself. They help to link cells to one another to form tissues. Look over here. You can see that these protein fibers of the cytoskeleton are actually going to pass through the plasma membrane of this cell, through the plasma membrane of the adjacent cell, and they're going to help to tie them together to help to build tissues. And then again, as we've said, the filaments, microfilaments, help to provide for movement of certain cells. I should also point out here that these things move up into the microvilli of cells and help to make those microvilli stiff. And so they're not quite as floppy as cilia would be. All right, another non-membrane bound organelle would be the ribosomes. And I want you to notice that the ribosomes are made up of two subunits, a larger subunit and a smaller subunit. And so when you draw a ribosome for me, don't just make a dot on a paper, draw the two subunits. Some people like to think of these as kind of an upside down acorn. Here's the cap of the acorn, and then here's the larger part of it. We'll talk later about the importance of these three binding sites that you see here that are labeled as E, P, and A. And when we talk about translation or protein synthesis, those will become important, and we'll do that later. These ribosomes could be found free floating around in the cytoplasm, or they could be attached to the rough endoplasmic reticulum. And again, as I've said, two subunits, a larger subunit and a smaller subunit, but both are composed of ribosomal RNA and then also protein that's been made in the nucleolus. And as we've said several times, there's no phospholipid bilayer that's associated with this ribosome. And then again, they are the sites of protein synthesis. We'll look at a quick overview of protein synthesis, but we'll go into more detail later on in class. Here's a ribosome. It might be attached to endoplasmic reticulum, or it could be free, and it has to work with messenger RNA that was made inside the nucleus and has since moved out into the cytoplasm. And then here is transfer RNA. Transfer RNA is going to bring amino acids in to the ribosome, and at the ribosome, these amino acids are going to be linked together to make a longer and longer and longer polypeptide. And it may get long enough to actually be a protein. But the messenger RNA is going to move through the ribosome, and more amino acids will be brought in here. Here's another one that just shows you slightly more detail, and we'll talk about translation in more detail in class. But again, here's the messenger RNA that was made in the nucleus. Here is transfer RNA that was also made in the nucleus, and there's the amino acid that it's bringing in to add to the growing polypeptide chain. Here you can see ribosomes that are on the surface of the endoplasmic reticulum. I do want to point out to you that the endoplasmic reticulum is membrane bound, and what you find right along here is phospholipid bilayer, even though they aren't showing it to you. And what's going to happen here is this completed protein is in the end of the rough endoplasmic reticulum. And because of the fluid nature of the membrane, it's able to pinch off and form this little three-dimensional bubble, which is known as a vesicle. Here you can see a little bit of detail of the rough endoplasmic reticulum. And again, I hope you remember that we said that the membrane of the rough endoplasmic reticulum is continuous with the outer nuclear membrane. And there you can see that right there. That's going to be true in more than just one place, even though they just show it to you here once. And then here is the phospholipid bilayer of the rough endoplasmic reticulum. And it almost looks as if this is an endoplasmic reticulum envelope, but we don't refer to it that way. These are flattened tubules. This is like a rather large tubule that's been flattened out, and you can see that it is indeed curved a little bit, and then it's studded with the ribosomes to make it into rough endoplasmic reticulum, also usually known as rough ER. Here you can see smooth endoplasmic reticulum that we'll talk about in the next video. Down here, what you can see, again, as we've already talked about, the little piece of rough endoplasmic reticulum pinching off and forming a vesicle which has protein in it. That's going to move over and merge with another membrane-bound organelle, the Golgi apparatus. And again, this pinching off and this merging of membranes is due to the fluid nature of plasma membrane. And so again, rough ER is involved in and works with ribosomes in synthesis of proteins. These proteins are either secreted to the outside of the cell through a process known as exocytosis. They could be used in membrane construction. Remember that a plasma membrane is anywhere from about 40 to 55% protein, and so these proteins could be incorporated into the membrane and become channels or transporters or enzymes. That brings us to the end of the second video on cell physiology. In the third and last video, we'll finish up talking about cytoplasmic inclusions and organelles, primarily organelles.